Hello students, now we will start with new topic, that is transcription. Transcription, also called as heterocatalysis. What does it mean? We know that DNA synthesizing new DNA is called DNA replication. We also gave a term for that as autocatalysis. Now, if the DNA is synthesizing an RNA molecule, means DNA is not synthesizing the same type of DNA, but it is synthesizing another type of genetic material, that is RNA, so called as heterocatalysis. DNA synthesizing RNA, this is other type of genetic material. This is called as heterocatalysis. Now, transcription, it differs in case of prokaryotes and eukaryotes, the total process and also the enzymes that are involved here. DNA synthesizing RNA, RNA synthesizing the proteins. This total process is called as central dogma. Here we are including the term transcription. Here we are including the term translation. The piece of DNA that is involving in the total central dogma is called as cistron. Basing on the term cistron, what type of cistrons are included in prokaryotes, what type of cistrons are included, means the number of cistrons present in eukaryotes and prokaryotes. In case of prokaryotes, the heterocatalysis occurs at different places, means simultaneously many number of RNA are synthesized at a term, so that many number of proteins or many types of proteins are also synthesized simultaneously. So in prokaryotes, the cistron is polycistronic. Here we see synthesis of many proteins, many types of proteins. In eukaryotes, the cistron is monocistronic at one time. The cistron is monocistronic, means at a time only one type of protein is synthesized and later another type of protein is synthesized. But all types of proteins are not synthesized at a time. One type of protein is synthesized at a time. Later, another type of cistron will include the process of total transcription, translation, and later another type of protein is synthesized later, means one after the other. This total transcription process includes the involvement of DNA and different types of enzymes and different types of DNA sequences or gene sequences. Transcription includes DNA double strands, next promoter sequence, next RNA polymerase, sigma factor, rho factor. These are considered as the regulatory proteins, and also Terminator sequence. How we have written here, promoter sequence, like the terminator or terminating sequence. Next, here we also read some of the terms like upstream and downstream also. Terms. DNA strands are of two types, that is coding strand, which gives its own copy for synthesis of mRNA. It is also called as sense strand. Now, another one is template strand on which mRNA is synthesized, and template strand also called as antisense strand. Template strand nitrogen based sequence are complementary to that of newly formed RNA sequence. Template strand, This is also called as antisense strands. This is complementary to newly formed RNA or complementary to mRNA. This coding strand gives its copy to mRNA. What about this promoter sequence? Promoter sequence, it is in eukaryotes. It is made of 25 to 35 base pairs. Now, in eukaryotes, it is called as Tata box. In prokaryotes, it is called as Pribno box, or this is also called as TAT, TAT box. Now, RNA polymerase, it is nothing but an holoenzyme. What is meant by holoenzyme? Means the enzyme that has both protein part and non-protein part. Means it has protein part plus non-protein part. Both of them is called as holoenzyme. The protein part of the holoenzyme is called apoenzyme. The non-protein part, it can be a cofactor, an organic cofactor, or an inorganic cofactor. Means nothing but the minerals. Now, this holoenzyme, it is having few subunits. Has 5 subunits. What are those? 2 alpha subunits. These alpha subunits help in mRNA chain initiation and interaction of regulatory proteins like sigma factors. It helps in mRNA chain initiation and binding of the regulatory proteins. Now, another one, beta subunit. That is only one. It also helps in chain initiation and chain elongation. Nothing but polymerization. What chain? mRNA chain initiation and elongation. This elongation term we also use with polymerization term also. Now, another one, that is beta prime subunit. This is considered as the largest subunit of RNA polymerase. So it helps in DNA binding, with RNA polymerase. Means RNA polymerase will go and bind to one specific area of the DNA at this beta subunit area of the RNA polymerase. This helps the sigma factor to recognize and bind on to the RNA polymerase. Sigma factor to bind to RNA polymerase, also called as core enzyme also. RNA polymerase. RNA polymerase also called as core enzyme. Core enzyme. We can also read like core enzyme has all these 5 subunits. 2 alpha, 1 beta prime and 1 omega subunits. Core enzyme can also be studied as having all these 5 subunits. Now sigma factor, which is responsible for initiation of the mRNA strand, it will just initiate and it will go off from the site of transcription. This rho factor, it is one of the protein part. It is a protein that binds to core enzyme or RNA polymerase for termination. For termination of mRNA. Next, Terminator sequence. Promoter sequence is nearly made of 30 to 35 base pairs. Terminator sequence is made of, this is approximate number, 20 base pairs, and this terminator sequence has more guanine cytosine nitrogen bases or base pairs. We can also tell as guanine cytosine rich area. Now, what is meant by upstream and downstream? If this is the area where mRNA is synthesized or transcription happens, suppose think that this location is the initiation place for mRNA. Means transcription starting site. We code this one with a numerical that is +1 because here there is no point of existence of RNA primer. So this can be written with a shortcut as T capital S means transcription starting site. Here I have given here plus symbol. Whatever the nitrogen bases or nucleotide sequences are present before that are considered as upstream area or upstream base space. So this is transcription starting site. Now in case of eukaryotes, this upstream area supports fitting or attachment of promoter at -35 base space area away from the transcription starting site. Means here is +1, -1, -2, -3. If we keep on calculating like that, at -35 base pair area, this promoter sequence will come and bind for the initiation of transcription. Now, so after formation of mRNA, at one place it has to stop its formation. Means that place will be terminator sequence. Whatever the nitrogen bases are present after terminator sequence, that place or that area is called as downstream area. Here also we can write like downstream area can be again +1, +2. It already started here with +1. If we want to change the numericals, it can be all plus symbols here in the downstream. But in upstream, all the numericals will be of negative sign only. That is before the presence of +1. Here in case of transcription, there is no point of including 0 place for the transcription area. It is -1. Immediately it will be -2. Because the total place is starting the addition of new nucleotides for the synthesis of mRNA. We will see some of the points regarding promoter sequence. Promoter sequence has 3 parts. One is core promoter. Second one, proximal promoter. Third one, distal promoter. Now, core promoter, it is considered as the most important one or considered as the start codon. Is very near to start codon. Next, RNA polymerase 2. Because we have in case of eukaryotes 3 types of RNA polymerases. RNA polymerase 1, 2 and 3. RNA polymerase 1 is responsible for synthesis of rRNA. RNA polymerase 2 is responsible for synthesis of mRNA. RNA polymerase 3 is responsible for synthesis of tRNA. There are some other types like 5S and that we will discuss later. But here, RNA polymerase 2 is involved in synthesis of transcription. Means a new mRNA. So, RNA polymerase 2 binds here. Next, this core promoter, also termed as Tata box in eukaryotes, This is also called as Goldberg-Haugnes box. This is present in, or this data box will write, is present in eukaryotes and archaebacteria. Then what is in case of prokaryotes? In prokaryotes, it is called, this core promoter is called Pribno box or TAT box. Now, this Pribno box constitutes of 6 nucleotides. Then what about promoter or proximal promoter? Proximal promoter has 250 number of base pairs, and this area is majorly responsible for attachment of sigma factor. It has 250 base pairs and sigma factor recognizes the site. Then what about distal promoter? It is also called as silencing gene expression area. So it enhances and silences gene expressions. Now, this is the area where regulatory proteins are binded. Regulatory proteins are binded. So this is about promoter sequence which binds at the upstream area of the transcription place. We will see the structural arrangement of transcription area. These are the double strands of DNA. So, one strand we can give term like coding strand. Coding strand is read from 5' to 3' area, which gives its own gene sequences to mRNA. If coding strand, if this end is 5', then it will be 3' of the other strand, that is template strand, and this will be 5'. You will write one point here. Coding strand is read from 5' to 3' direction. Next, template strand is read from 3' to 5' direction. Now, if a question was asked, what is the streaming means type of stream that is present on 5' end of coding strand, then it will be upstream. This is template strand. Think that this is the area where transcription starts. So this is called as transcription starting area. Some 1, 2, 3 and so on like this at -35 nitrogen base or nucleotide area. Here will be attached the promoter sequence. Promoter sequence. This area is called, so we can write this one on like this. You can write here -35 here, which initiates of the promoter sequence. This area is called, not specifically this total area is called upstream area of transcription starting site. Now, where does this RNA polymerase binds? RNA polymerase can be binded by occupying all this area which I am marking with red color marker. So here the RNA polymerase has also occupied the transcription starting site. RNA polymerase 2 has a special nature of initiating or adding the nucleotide to itself because it doesn't need the RNA primer for addition of new nucleotides. So in case of transcription, there is no point of existence of RNA primer. Now, so this is RNA polymerase 2. Here there is no sigma factor, so we can also give a term like core enzyme. Somewhere here, after some distance, will be terminator sequence or terminating sequence. It is made up of approximately 20 number of nitrogen bases or nucleotides. Now, this terminator sequence has stop codons. Here, to this RNA polymerase binds a special type of factor that is called as sigma factor. Sigma factor is used only for initiation of the mRNA strand. Here will be present the rho factor. So, when it is near to the terminator sequence, when rho factor binds, it means that mRNA is stopping its formation. This RNA polymerase will scroll over the template strand for synthesis of new mRNA till it has reached the terminator sequence and then binded with rho factor. So, here will be the new mRNA strand that is formed. So, this can be new mRNA. New mRNA is always termed with a term like hnRNA, means heteronuclear RNA. We will see about all this during the process of a transcription. So, these are the main points. So, we can also write one more point like here GC rich area, guanine and cytosine rich area. So, this is about the areas that are present. Here we have written upstream area. So, this area can be called as downstream area. Of the transcription unit. Now we will see the process of transcription. In the process of transcription, the DNA double strands are involved. Here, mRNA is formed on the template strand, means all the nitrogen base template strand. All the nitrogen base pairs are complementary to the nitrogen base pairs of the template strand, and whatever the gene sequences are there, those are nothing but a copy of the coding strand. Whatever gene sequences are there, as it is, gene sequences will be present on mRNA, but the only difference will be instead of thymine, there will be uracil in mRNA. Means coding strand is giving its copy and template strand is helping in formation of mRNA. As we have discussed before, what are the included substances in the mRNA? Double-stranded DNA. Now, 5' to 3' end directed strand is coding strand, which is also called as sense strand. Now, the promoter is somewhat away from the area where transcription has to begin. So, this can be called as transcription starting area, which we indicate with +1. So, a little bit away from that. In case of prokaryotes, it will be -10 base pairs distance away from the TSS, means transcription starting site. In case of eukaryotes, it will be -35 base pair distance away from the transcription starting site. Now, RNA polymerase will come and recognize this prokaryote promoter sequence on the means double-stranded of DNA, and it will bind there. It is not occupying only the promoter sequence, but also the transcription starting unit also. So, here the new mRNA starts forming. Now, the mRNA always will be having the initiating codon like AUG on its first codon. Then, what type of complementary strands should be present on template strand of the double strand? If it is adenine, then it should be thymine, adenine, and cytosine. See here, in case of thymine, there is uracil in mRNA. So, it is considered as the initiating codon. It starts at transcription starting area. For that purpose, the sigma factor will come and initiate the sequence that is AUG codon sequence of the mRNA, and it will get separated off. Sigma factor function is just binding to the RNA polymerase, initiating the mRNA synthesis and disappearing from that area. So now AUG codon has started here at the transcription starting site, and now the sigma factor will put off from there. Next, in all these cases, if we observe, template strand is read from 3' to 5' direction, coding strand is read from 5' to 3' direction. Even mRNA also, it is beginning its formation from 5' end only of the mRNA towards the 3' end of mRNA. Now 5' end will start releasing from the template strand slowly as the thread length increases. So this end can be 5' end of mRNA, that is complementary to the 3' end of the template strand, and it keeps on elongating. RNA polymerase, when it binds onto the double-stranded DNA, first of all, it unwinds the double-stranded DNA and gives a space for new synthesis of mRNA, so that wherever RNA polymerase is diagrammatically represented here, I have drawn a distance between the two strands, here and here also. Means it kept on after sigma factor got disappeared, then RNA polymerase alone started scrolling over the DNA strands and unwinding the DNA strands and supporting in synthesis or addition of new nucleotides for the synthesis of mRNA. So this is mRNA. Its other end will be 3' end. Now, this process has been kept on adding of nucleotides. Now we will give the three different terms that are included under transcription. Till initiation here of the mRNA strand, we can give a term like steps are. One is initiation. Second one, here I have told you new nucleotides for synthesis of mRNA kept on adding by the presence of RNA polymerase. RNA polymerase is having a nature of no need of RNA primer for the addition of nucleotides. So this step is, second one is elongation, is done by RNA polymerase alone. There is no existence of sigma factor here. Third one, see this diagram. So here 5' end of mRNA, 3' end of mRNA still attached to one end of the template strand. Now it has reached a sequence called as terminator sequence. Terminating sequence. mRNA will be formed till it has reached the terminating sequence. The 5' end is released on the 3' end of the template strand or on the 5' end of the coding strand. Wherever this RNA polymerase has entered, there will be unwinding of the double-stranded DNA. But now here, in this diagram, it has reached the terminating sequence. So the last step will be termination. So initiation is because of this sigma factor which has binded to RNA polymerase. Now, in the terminator sequence, now the rho factor will be binded, which is indicated with capital R, small h and small o. When this binds, then RNA polymerase stops the polymerization activity and it will put off from that place. Now, mRNA that has formed newly includes different types of gene sequences. Some of the gene sequences which are responsible for next protein translation, those are called as exons. The line-like structures which I have drawn here are not coding for any next coming translated proteins. Those are called as introns. Means coding sequences, non-coding sequences together will be involved in the newly formed mRNA, which is called as heteronuclear RNA. So finally, we have finished in case of transcription process 3 important steps, that is initiation because of the sigma factor which is present attached to the RNA polymerase, and elongation alone by the RNA polymerase, termination with help of rho factor which is binded to RNA polymerase when it has reached the terminating sequence. The RNA polymerase, when it has reached terminating sequence, at the point of terminating sequence, there are 3 different types of stop codons. What are they? So here we can write the stop codons at terminating sequence are 3. What are those 3? terminating sequence, terminating codons are nothing but UAA, UAG, UGA. UAA is also called as ochre. UAG is also called as amber. UGA is called as umber. This we also call as opal. But now the question, where are these stop codons present? Are they present on the mRNA or on the template strand? So these stop codons are present on, means coded on mRNA. Coded on mRNA. It means that the template strand will be having some sequence like for UAA on mRNA, the template will be having adenine, thymine. For UAG of mRNA, it will be having adenine, thymine, cytosine. For UGA of mRNA, the codon sequence on template strand will be adenine, cytosine, thymine. But on coding strand, what are the types of codon sequences? On coding strand, the sequences that must be copy of this one means thymine, adenine, adenine on the coding strand, thymine, adenine, guanine on the coding strand, thymine, guanine, adenine on the coding strand. So these three, ATT, ATC, ACT, are responsible for coding the stop codons of mRNA. So this is about till the formation of termination step in the transcription. Now we have got here one heteronuclear RNA. We will see about that. HNRNA means heteronuclear RNA. This is also considered as newly formed inactive form of mRNA. Form of mRNA. Now, it has non-coding sequences called as introns. These do not translate for proteins, and coding sequences which involve in the process of translation, those are called as exons. Sequences, those are called as exons. Now, if we go for diagrammatic representation, we will shade the exons in thick diagrammatic representation. So we can label this one as exon, this thin one as introns. As we have said, introns are not coding for anything. Then what is use of existence of introns? Introns are not completely the useless particles. Those are nothing but a combination of nucleotides, but here they are not used for translation. So in eukaryotes, since DNA is having coding and non-coding sequences, so that we have got mRNA like this. In case of prokaryotes, the DNA sequence contains completely coding sequences only. There is no point of non-coding sequences in prokaryotes. So there we get continuously exons only in prokaryotes. In eukaryotes, there is an enzyme called spliceosome. Spliceosome. This spliceosome is nothing but an RNA protein complex. It is helping in breaking off. It helps or involves in breaking or cutting off introns. We can also use the term separating of introns. So that what happens, spliceosome is removing the introns, so we get a combination of exons only, like this. But exons has to be attached one with each other. So in eukaryotic body, there is another enzyme, that is ligase. Ligase is also called as binding or joining enzyme. So when ligase enzyme is involved, we are getting a continuous strand of mRNA, means active form of mRNA continuously. Ligase is a binding or joining enzyme. Now this is completely formed mRNA. But still mRNA is considered to be inactive if its structure is like this. On one end, that is on 5' end of mRNA, methyl groups are added. We give a term like this as, this end is called as cap. So cap of mRNA is of methylated guanine. At one area, there is non-coding some sequences. Later, there will be start codon. That is, AUG sequence will be there on mRNA. Next, some of the genes are arranged as structural genes. All these are nothing but from the exons only. After the structural genes, again, there will be stop codons, which we have written here before. UAA and UAA, that is Walker, and UAG, that is amber, and UGA, that is amber. Later again, non-coding sequences will be there. From here onwards, there are addition of 200 to 300 number of adenines. So this area is called polyadenylated tail, and the process is called as polyadenylation. So in case of one active completely formed mRNA, we have 5' methylated guanine cap. Next is non-coding sequence. Next is AUG, initiating codon. AUG is initiating codon. It initiates for methionine amino acid. So now we have seen complete formation of mRNA by the process of transcription. Here we see some of the terms like split genes in NCERT. What are split genes? Split genes means one intron between two exons. One intron between two exons. That is called as split gene. So here exon, intron, exon. This total combination is called as split gene, and this arrangement of split genes like this is considered to be one of the similarity to the ancient people or ancient gene arrangement. Now, in NCERT, we also have another term, that is reminiscent of antiquity. Reminiscent of antiquity. What does this mean? It means that presence of introns is considered as similarity of ancestral gene characters or characteristic features. Similarity to ancestral genes. That is meaning of reminiscent of antiquity. And this is about the transcription process.